across all the viruses we tested for. Well, there's so much going on within each study. I suppose the ideal thing, I think the holy grail would be to find a particular sequence that is seen in children with apnea or very severe RSV, and see if we could develop an assay for that. That would be the holy grail, because then we could say, you know, this child may be small, may have risk factors for becoming apneic, but based on the RSV sequencing, we can say this one is not at risk. Or alternatively, we can look at another kid who looks perfectly well, you know, minor bronchiolitis, who you'd like to send home, but you know that the particular strain he has is associated with apnea. And that will be useful information. We're a bit of ways from being able to get there. We may not get there. It's still a hypothesis, and we're still working on testing it. The importance of this from a researcher's point of view, and ultimately from a clinician's point of view, because these tests will ultimately become more and more routine, is that when I see a child now, he gets an antigen test, and he gets it for RSV, and that's about it. If I want to do PCR, I have to freeze the samples to -70 and transport them on dry ice. And in a busy ER, in a hospital with our level of resources to spend per patient, that's not going to happen outside of a research project. Ultimately, if we're going to use these tests, we're going to have to find a way of being able to ship them at room temperature in a medium, in a tube. And that's where these things come in handy. And that's where this is going, so that ultimately we will be able to say, we don't have to refrigerate everything, we don't have to freeze it, we can simply take the swab, put it in this, and send it to the lab. And that's the value of this kind of technology in terms of swabs and transport media.
Okay, my name is Dr. Martin Adelson. I'm the Vice President and Director of Research and Development of Medical Diagnostic Laboratories. Medical Diagnostic Laboratories, or MDL, was founded back in 1997 by Dr. Ellie Mordecai, and at the time the focus of the laboratory was on tick-borne diseases. We're in the area of northern New Jersey, central New Jersey. There's a large amount of tick-borne diseases here, including Lyme disease, Babesia, Bartonella, which we've done some basic research on and published, ehrlichiosis. And the focus of the laboratory was on molecular diagnostic testing and also on serological diagnostic testing for such states. One of the hallmarks of MDL that's been very successful for us as a business is that we have developed a platform like OneSwab, which is used for the collection of cervical vaginal swabs from women, like EuroSwab that's used for the collection of urine then from men. And we've taken this transport media, which can be delivered to the laboratory, and we've associated a large number of tests with them. Very common tests like Neisseria gonorrhea, chlamydia trachomatis, very esoteric tests for, say, lymphogranuloma venereum then for men. We've associated different HPV assays, molluscum contagiosum viruses, different pathogens that are associated with bacterial vaginosis, and we've provided the physician with the flexibility to do as few as 1 or 2 tests, you know, if it's routine screening or if they're presenting with some very unknown symptoms and the physician really suspects that it's something rare, we have the flexibility to provide that information to the physician as well. We've recently changed our laboratory workflow around in the laboratory. We've implemented more of a semi-automated robotic system in the lab, and one of the hallmarks is we've been able to decrease the turnaround time of our results then to something that's in the range of 24 hours from when a specimen comes in the door to when the result leaves. And this is a huge advantage for us in the field. It's a very scalable system, and we're looking forward to now coupling this decreased turnaround time with the high sensitivity and the high specificity that's associated with the molecular diagnostic testing that we perform right now. Within the research and development arm of Medical Diagnostic Laboratories, we're focusing on several basic research studies. And among these, we're looking at the pathogenesis that are associated with different organisms, and some of them are pertaining to women's health. And when we started moving into that particular kind of field, we were contacted by Dr. Paul Walsh at Kern Medical Center. And at the time, he was interested in publishing and performing basic research studies on respiratory viruses associated with pediatric infectious disease. And this struck us as a very interesting concept then, because on the development side within R&D, we were interested in developing diagnostic tests that would be associated with pediatric infectious disease. We wanted to start to branch away, we wanted to start to supplement the testing that we were performing in women's health and also in urinary tract infections and add pediatric infectious diseases to it as well. And the initial study was very interesting to us because it would provide us with clinical specimens that we could use for test validation. And Dr. Walsh sent to us a large number of 600 or 700 samples then that were nasal washes. And we used these nasal washes then to develop tests specific for respiratory syncytial virus A, RSV-A, RSV-B, Bordetella pertussis, Bordetella parapertussis, and also human metapneumovirus. And the nasal washes were sent to us. We determined the prevalence, and Dr. Walsh went and presented this at 2 conferences associated with emergency medicine. Now, as the year went by, the initial study was very interesting to us, but we wanted to expand it. And we became interested then in the flock swab technology that was presented then by Copan Innovation. And this technology then, actually there's a flock swab that's right here, which were developed then. And these swabs, they have guards then, and they're specifically set at 25 millimeters in terms of length, and these would be used then in order to insert into the anterior nair of the infant. And they would be then put into this UTM transport media, this UTM-RT, and then sent to the laboratory for downstream testing. In the study with Dr. Walsh, when the specimens would arrive in the laboratory, it would go one of 2 routes. The first would be for DNA extraction, which used automated procedures that we have developed in the laboratory, and these would be used then for the testing for Bordetella pertussis and also Bordetella parapertussis. Separately, we used more manual RNA extraction procedures, and these were used for the testing then for the RSV-A, RSV-B, and also human metapneumovirus, influenza A, and influenza B. And we already had a large number of tests developed. We started a study then with Dr. Walsh this year in which we were looking at the nasal washes which he had initially been performing. And then we looked at if we took the nasal wash and then diluted it 1-to-1 with UTM-RT, and then also if we were to compare that then with the flock swab, which could be put directly into the UTM-RT. And these specimens were sent to our laboratory, and at this point he's sent maybe about 600 of them or so over the last flu season. And we tested them for human metapneumovirus, for RSV-A, RSV-B, Bordetella pertussis, Bordetella parapertussis, and also tests that we developed for influenza A and influenza B. Flock swab itself was one that we developed as the study was progressing, and this was through a collaboration with Dr. Paul Walsh and also Copan Innovation and ourselves. And some of the beauties of this swab then are the length of it. It's very short. You're able to get a lot of control then in collecting the patient specimen. There's a guard that's present on it, which would limit the input then into the nose of the infant then, especially as they're moving around. And then also the unique design of the flock swab itself, which enters into the mid-terminate region then of the nose. And one of the beauties of this as well is that it doesn't retain large quantities of material like more traditional swabs. It has a very high release rate in which when one puts it directly into the UTM-RT, it gets released from the flock swab into the media, and then this is taken for the downstream testing, which in turn can increase the sensitivity. The study at this point is still currently underway. We're still receiving specimens from Dr. Walsh. We have processed 98 samples so far with respect to those that were the nasal washes mixed with the UTM-RT and compared them against the flock swab that was put directly into the UTM-RT and then sent to our laboratory for either RNA extraction for the respiratory viruses or DNA extraction for the Bordetella pertussis and the Bordetella parapertussis. And the results that we've seen so far is that the flock swab in the UTM-RT is at least equivalent to the nasal wash at this point. We have detected a larger number of positive specimens that were confirmed by DNA sequencing for influenza B and also for RSV-A, but we're still processing the samples at this point. It's attractive to us because because if one compares then the flock swab then collecting from an infant that's coming to an emergency room and not in very good shape, and then compares that against the nasal wash, the flock swab is much more patient friendly, it's much less invasive, and it would be easier for the physician to collect and then send to the laboratory for analysis. From our initial data, it looks very possible too that it's more sensitive as well. And we've read the studies then, we've seen the reports then that the flock swab retains less specimen, it gets released very quickly into the transport media, and that in turn would increase the sensitivity of our assays. So we'll have more as the season finishes, which is almost upon us right now. The results of this study are going to be presented at the Clearwater Virology meeting, which is meeting at the end of April and then the first 2 days of May in Clearwater, Florida.